Cellular respiration. Cellular respiration is the reciprocal reaction of photosynthesis. Cellular respiration takes place in all organisms. It takes glucose, or our energy molecule, and breaks it down into usable energy for the cell. This is the overall equation for aerobic respiration. It is sugar, oxygen, going to form water and carbon dioxide, and in this process it releases energy, or ATP. This is a redox reaction. Redox means reduction, oxidation. And what that means is oxidation is a loss of electrons. Reduction is a gain of electrons. So the way to remember that is oil rig. Oxidation is a loss, reduction is a gain. The other thing that I want to go over before we get into respiration are electron carriers. Electron carriers are molecules that will pick up hydrogen electrons and they will shuttle it from one process to another. NAD is the oxidized form, NADH is the reduced form. FAD is the oxidized form, FADH2 is the reduced form. This is the overview of aerobic respiration. There are several steps that the cell goes through in order to take glucose and make it into usable ATP. We have two types of respiration. We have anaerobic and we have aerobic. Aerobic occurs in the mitochondria, mostly. Anaerobic occurs in the cytoplasm. Anaerobic occurs in all organisms, and this only occurs in some. Aerobic respiration is mostly in eukaryotic cells. There are some prokaryotic cells that can do a aerobic respiration, that they have specialized membranes in order to go through the process, that they don't actually have a mitochondria. Anaerobic happens with the absence of oxygen. Aerobic occurs with oxygen. Let's talk about anaerobic first. Anaerobic respiration happens in the cytoplasm, and it happens without oxygen. There are two steps. The first step of respiration is glycolysis, and this is the same for both types of respiration. The second step in anaerobic respiration is fermentation. The first step, glycolysis, is going to be the same between anaerobic and aerobic respiration. This is said to be the earliest, evolutionarily wise, energy pathway because it is done without oxygen. It's done in the cytoplasm, something that all organisms can do. If we take a look at glycolysis, we take glucose. Glucose is a starting molecule, and turns into two molecules of pyruvate or pyruvic acid. This happens in the cytosol or the cytoplasm. We need to put energy in order to get this going. So we put in 2 ATP, and then we get out 4 ATP. Here it says 2 because it's a net gain of 2. We profit 2 ATP. 2 molecules of pyruvate are made. If we look at the number of carbons, glucose has 6 carbons. If we cut that in half, it's going to make 2 molecules of 3 carbons each. Pyruvate has 3 carbons. We have 1, 2, 3, and 1, 2, 3. The other thing that we do put in is NAD. And we get out NADH. NAD is the limiting factor because this ATP can be used to replace that ATP. NADH, we need another way. NAD, we need another way to form that or make that, and that's where the second process of fermentation comes in handy. Let's review glycolysis. The input is glucose, which we get from our diet, 2 ATP to get it started, and 2 NAD. Our output is 2 pyruvate or pyruvic acid, 4 ATP, giving us a net gain of 2, and then 2 NADH. This is the process of glycolysis. Glycolysis is not just automatically cutting glucose in half. There are a series of steps that have to go through in order to go from glucose to pyruvic acid. You do not need to know these steps. You just need to realize that it is a series of steps. We have ATP going in, we have ATP coming out, ATP coming out. we also have our NAD going in as well. This ATP that we make can supply this. This NAD, we need some way to form that NAD, which is where fermentation comes in. We have two types of fermentation that we're going to talk about. We have alcoholic and we have lactic acid. Again, this is happening in the cytosol or the cytoplasm. The point of fermentation is to make our NAD, to keep glycolysis going. The first type of fermentation we're going to look at is lactic acid fermentation. During lactic acid fermentation, we are going to change our glucose into pyruvate, which is our glycolysis. And remember, we put in our 2-NAD and we get out our ATP. And then during fermentation, lactic acid fermentation, we're going to take pyruvate and break it down into lactic acid. We're taking the 2-NADH, we're making NAD, and that NAD can go back up to glycolysis to keep it going. Lactic acid fermentation can take place in bacteria and in our muscles. Us as humans have utilized the waste product of lactic acid. So this is a waste. We have utilized that in order to help benefit us. So we have used bacteria that do lactic acid fermentation to help us make things like yogurt or pickled vegetables. Our muscles form lactic acid when we're exercising and we go beyond a point of not having any oxygen available. The muscles can then flip into anaerobic respiration to keep themselves going. After sustained exercise and lactic acid is building up in the muscles, the next day that can be quite painful. So the pain in your muscles after exercise is that lactic acid buildup from the lactic acid fermentation. The other type is alcoholic. Again, we're starting off with glycolysis. We have our glucose, we break it down into our 2-pyruvate, we have our ATP come out, NAD goes in, NADH comes out. We're going to change that pyruvate into carbon dioxide and alcohol. Don't worry about this acetaldehyde step, we're not concerned with that. So we're going from pyruvate to alcohol, ethanol is an alcohol, and carbon dioxide. The important molecule here is our 2-NAD. Those 2-NAD are going to go back to glycolysis to keep it going. Alcoholic fermentation can be found in some bacteria as well as yeast. We have taken the benefit of this from the yeast and using yeast to help us make things like bread. The carbon dioxide that is produced helps bread dough rise. And also for beer, wine, and spirits, we feed yeast a sugar juice. They eat that sugar, break it down, go through alcoholic fermentation, and produce alcohol. Aerobic respiration happens in the presence of oxygen. Aerobic respiration has 3 steps. Well, 3.5, 4. Our first step is going to be glycolysis, and remember, glycolysis is the same. Then we have the citric acid cycle, and then we have the electron transport chain. Our first step, glycolysis, is the same. We take glucose, break it down in half to pyruvate. We get our 2 ATP, our 2 NAD. So we get our 2 pyruvate, 4 ATP, net gain of 2 NADH. This NADH is going to go to the electron transport chain in order to help power the formation of more ATP. The next kind of the half step that I was talking about is pyruvate oxidation. We're going to change pyruvate once it reaches the mitochondria before it goes into the citric acid cycle. Pyruvate is going to change into acetyl-CoA. We're going to lose one of the carbons in this process and form a molecule of carbon dioxide. We're also going to make some more NADH. Again, this NADH is going to go to the electron transport chain, and this carbon dioxide is the carbon dioxide that we breathe out. So our next step is the citric acid cycle. The citric acid cycle is also known as the Krebs cycle. During this, we have acetyl-CoA go into the cycle, and again, you don't need to know all of the in-between steps, you just need to know the overview. The reason why it's called the citric acid cycle is because citric acid is made. Citrate is the same thing as citric acid. We have a formation of NADH here, here, and here. We have the formation of FAD H2, our other electron carrier, and that is there. We have the formation of some ATP, which is here. And then we also have carbon dioxide as a byproduct, here and here. Again, you don't need to know the in-between steps. You just need to know that the citric acid cycle starts with acetyl-CoA and ends with NADH, FADH2 going to the electron transport chain, ATP being formed, and CO2. The way that ATP is formed during the citric acid cycle and glycolysis is called substrate-level phosphorylation. You'll notice that the substrate-level phosphorylation produces less ATP than the oxidative phosphorylation, which happens in the electron transport chain. The citric acid cycle takes place in the matrix of the mitochondria. After the citric acid cycle, we're going to go on to the electron transport chain. The electron transport chain happens in the inner mitochondrial membrane. So here is the inner membrane. In the inner membrane, we have some proteins. You do not need to know the names of those proteins. You just need to know the proteins are there. This is the inner mitochondrial space up here, and down here is the mitochondrial matrix. So here is where our citric acid cycle is taking place. From that, we have the NADH and the FADH2. And what happens is the NADH and the FADH2 drop off their hydrogen. That hydrogen is a proton and an electron. The electron portion of it, the proteins within the membrane have an affinity for that, and each protein has much more of an affinity for that electron, and it pulls that electron down the electron transport chain. Last acceptor is oxygen. So oxygen is the final electron acceptor on the electron transport chain, and this is where water is formed. The proton from the hydrogen crosses the membrane and builds up a gradient in the inner mitochondrial space. As it's building the gradient, it wants to go from an area of high concentration to low concentration. There's too much hydrogen ion building up in that space. And it looks for a way to get back to the mitochondrial matrix. The only way it can get back is through an enzyme that makes ATP. Here is the electron transport chain. Our hydrogen gradient's building up here, and the only hole that's available is the ATP synthase. It's an enzyme. The enzyme sits in the membrane and is powered by the movement of hydrogen going through that enzyme. Hydrogen go down through the middle of ATP synthase and spin it around, which powers it to form our ATP. The movement of hydrogen ions going from the inner mitochondrial space to the mitochondrial matrix is called chemiosmosis. This whole process is oxidative phosphorylation. We're making more ATP. This process makes more ATP than our substrate level phosphorylation. Let's take a look at how much ATP is formed per 1 glucose going through the entire aerobic process. We have 1 glucose turning into pyruvate in glycolysis. So glycolysis provides us with 2 ATP directly and then 5 ATP from our 2 NADH that later go down to the electron transport chain. The pyruvate decarboxylation, or the pyruvate oxidation, gives us 5 ATP from the 2 NADH that it forms. Our citric acid cycle directly gives us 2 ATP, but then from the FADH2 and the NADH that are formed, we get 18 total. If we add all these numbers up, it comes up to 32 ATP. You may read in some places that it goes up to 38 or 40. That's the theoretical value. Theoretically, more ATP should be made per glucose. But with all of the chemistry that actually goes on and the way that some of the reactions occur, less ATP is actually formed. It's somewhere between 30 and 32. If we compare aerobic respiration to anaerobic respiration, we can see that anaerobic respiration is very quick. If you need quick energy, anaerobic is the pathway to go. Aerobic takes much longer to go through that process, but it's much more efficient. You get so much more ATP per glucose going through aerobic respiration than you do anaerobic. Anaerobic respiration can be done by virtually all organisms. Aerobic respiration can only be done by some, and that is cellular respiration.